right, good morning, everybody. Wow, what an honor to be here. And I'm very excited to tell you a bit about resurrection plants today. And most of what I'll focus on is about opportunities and potential innovations that could be derived from these amazing plants. But I do want to start with a small dose of reality. So, as I'm sure you all know, our planet is changing. And drought, in particular, is increasing in frequency and intensity. And many regions of the world are becoming increasingly arid. As water becomes scarcer, ecosystems are collapsing and crops are failing. Species are disappearing, and this threatens the stability of our natural world as well as our global food security. But what if nature has already engineered a solution to this threat? Imagine a world where organisms could come to a standstill in periods of scarcity, for months, even years, as they wait for conditions to become favorable again. Now, imagine if we could harness this ability to address some of society's greatest challenges. This is the humble goal of my work. In some of the world's harshest environments, life has evolved in astonishing ways. There are organisms that can survive being completely dried out, seemingly suspended in time, but still alive. We call this desiccation tolerance, and it's one of nature's most extraordinary phenomena, enabling survival in some of the world's harshest conditions. And perhaps the most famous of the desiccation-tolerant organisms are resurrection plants. These plants desiccate so completely that there's not even a single monolayer of hydration around cellular macromolecules and structures. But they are still alive. They enter a state of quiescence or dormancy where they appear dead, but they spring back to life with the first summer rains. And as you can see, witnessing their revival is like watching a small miracle unfold. Resurrection plants survive in some of the most extreme, remote, and awe-inspiring landscapes on Earth. They can be seen hanging dangerously off the sides of cliffs, on sweltering rock surfaces with little to no soil, and on barren desert slopes. And I've had the great privilege to visit many of these beautiful landscapes, from the high plains of Cerro de Sipo in Brazil to the rugged Kopi and Ruari of Limpopo, South Africa. And these landscapes are humbling in their sometimes desolate vastness. And these plants, in their defiance of nature, I think are a true testament to evolution's ingenuity. But we're not just marveling at these plants, we're actually studying them, and we're trying to understand how they accomplish this resurrection, this very remarkable feat of survival. We use comparative methods to look across diverse resurrection plants to try to identify core or shared mechanisms of tolerance. Good. The slide didn't look good last time. So this actually involves zooming in from various different perspectives, from their unique evolutionary history, to their intricate physiology and cell biology, to their complex genomic architecture. By taking this comprehensive or systems biology approach, we're trying to get a holistic view of how these plants survive desiccation. And there's actually a long history of studying and being aware of desiccation tolerance. And one of the key discoveries that was made quite early on is that when resurrection plants dry out, they actually vitrify their cells to form this sort of bioglass, which stabilizes the cellular components, protecting them from the stresses of shrinkage and molecular crowding that happen during drying. But recent work has taken this one step further to show that intrinsically disordered proteins, these fascinating flexible molecules shown here, are actually critical to giving these bioglasses their strength and ensuring protection in a dry state. Another important discovery that we've made recently is the identification of a specific family of genes called ELIPs that play a major role in desiccation tolerance by protecting chloroplasts from light damage. These genes are actually expanded in the genomes of all known resurrection plants in tandem arrays, pointing towards their critical role in desiccation tolerance. And what I think is particularly interesting here is that this expansion of these genes has happened independently and convergently in many diverse species, highlighting that nature has actually engineered desiccation tolerance time and time again. But hey, our research isn't just curiosity-driven. We're actually trying to understand how we could harness these innovations for some of our own uses to benefit society and the world. And to be clear, understanding the mechanisms of desiccation tolerance has enormous potential for practical applications. Take, for example, the technique of zero preservation, or the dry storage of biological materials. So these techniques are actually already being applied to biological macromolecules like RNA vaccines. And this is eliminating the need for refrigeration and costly cold chain logistics, which has the potential to democratize access to life-saving medicines and vaccines around the world. But imagine if we could scale this up to preserving whole cells and complex tissues. That could enable the preservation of recalcitrant seeds, of explants, of embryos, and other important biological tissues, which could revolutionize biodiversity conservation, allowing us to safeguard the germplasm of endangered species and crops for future generations. Taking it one step further, imagine if we could apply these techniques of zero preservation to whole organisms. Now, I know it sounds and looks a little bit like science fiction, but biostasis, or the long-term preservation and suspension of life, could become a reality. And this, I think, obviously would have profound implications in medicine and agriculture, and even in things as futuristic as interplanetary travel. And of course, there's the obvious fact, and maybe the reason we're all here today, that desiccation tolerance could completely change the game in agriculture. With drought becoming more intense and more frequent, many communities are struggling with crop loss and global and food insecurity. And in order to protect against this, we need crops that can withstand unpredicted and extreme events. And so this has actually been a long-standing goal of desiccation-tolerant research to improve crops and make them more drought-tolerant. And we're working quite hard to identify the key genetic features that enable desiccation tolerance, like ELIPs, which I mentioned earlier. And we believe that by inserting cassettes of these key genes under the control of stress-inducible promoters, that we could actually improve drought tolerance in crops. But it's a long-standing goal, and there are a number of challenges. So there's actually thousands of genes that are involved in desiccation tolerance. They're controlled by complex regulatory networks, and you may know, if you're a plant scientist, that plant transformation is notoriously fickle. But still, this is an important goal because the potential to safeguard our food supply is enormous. Now, the microbiome of resurrection plants is another extremely promising area of research, and we've actually already had successes with small-scale interventions and microbial inoculants derived from resurrection plants. This is the first step to developing biostimulants, which could be applied to agricultural systems to improve drought tolerance without the need for genetic modification. However, to achieve these lofty goals, we really do need to accelerate scientific breakthroughs. And to do that, we need to change how we work together. So right now, research on desiccation tolerance is conducted in siloed subdisciplines, as shown here on the right, on your left. Different groups study different organisms at different scales, and there's really insufficient integration and information exchange, which is hindering our ability to generate breakthroughs and conclusive understanding. Perhaps an even greater challenge that I think is especially relevant today is that science itself is unequal and quite exclusive. So the global south, where much of the world's biodiversity lies, is sadly often left out of the conversation. And this is a remnant of colonialism, which historically shaped who had access to knowledge, resources, and opportunities. And sadly, these patterns still exist in science today. But let's flip the script. Let's bring together scientists from all over the world, especially those who have been historically marginalized, and try to work together to harness the power of plant resilience. Imagine what we could accomplish with more voices, more perspectives, and more hands at the table. And this is what we're trying to do now through grassroots collaborations and initiatives like WALI, the Water and Life Interface Institute. We're building a global network of scientists studying desiccation tolerance across scales. We're breaking down silos, trying to integrate knowledge, and work together to find solutions that are locally relevant but globally scalable. And it is through these types of inclusive collaborations and an empowerment of people from all walks of life that I believe we will achieve true innovation. So, in closing, as the world changes, I'd argue that we can look to nature for a blueprint of survival. Desiccation tolerance and the resurrection plants that embody it offer us a small glimpse of what's possible. But it's up to us to come together to harness this power of plant resilience and address some of society's most pressing issues. And it is my great hope that today's conversation and the ripples of it in the future accelerate this goal. Thank you so much for your attention.